and I want to thank Olga and everyone else for their hospitality here. So what I'm going to talk to you about is our effort to understand how cells compute and how we can harness that understanding. So as Olga said, I have been in this emerging field of synthetic biology. Really, the problem that I've been interested in for many years is the question of how living cells make decisions. That we have, what distinguishes living systems is that they have the ability to sense what's going on in their environment and then can make very complex decisions. And somehow systems of molecules, are genetically encoded molecules, give cells this kind of capability. As we've studied this and sequenced the genes, the genomes of various organisms, one of the things that emerged was that there were, in many cases, kind of common molecular building blocks or modules that were found to underlie a lot of the signaling networks. And so in the last maybe 5 or 10 years, we have kind of converted from trying to dissect and break down complex pathways to trying to reframe the question in a different way, which is how can we take simple molecular building blocks and start to build cells that carry out complex functions? And that really, this effort to try to learn by building is what characterizes at least the synthetic biology that we do. So how did evolution take these sort of building blocks and build cells that can do the kinds of complex behaviors that we see? And so a lot of this is really aimed at trying to elucidate what are the design principles, are there sort of common themes and motifs that are used to achieve certain kinds of behaviors like homeostasis, other things like that, spatial organization. But one of the things that in getting into this that we realize is that if we really understood these principles and we could actually forward design biological systems, that it would be incredibly useful if we could design cells that carried out new tasks that were, say, therapeutically useful, but that had the same kind of precision that living cells, evolved cells have. And so that's really what I'm going to focus on today. And before I get into what we've been doing, I just want to kind of contrast. The basic idea here that I'll be talking about is about trying to use cells, living cells, as therapeutics. And I just want to contrast that with our, sort of the common modalities that we use to interface with the body and disease, which is typically small molecules or biologics like antibodies or hormones or other things like that. And so usually in these cases we have a sort of molecular entity that we put into the body. It goes everywhere, and it's largely will recognize targets based on sort of a defined molecular surface area. And these can be obviously very, very effective, but there are limits to sort of how much specificity and how precise these can be. And they go everywhere in the body, so they almost always have systemic side effects because molecules are used over and over again in the body in different places. And in contrast, if we think about like an immune cell, which is shown here, which is in a way an evolved therapeutic agent, this is a human neutrophil. This is a living device that is, in this case, sensing molecules that are given off by that little bacteria, that little dot. And those actually cause these major changes in the shape of the cell, and they are calculating kind of where it is, and then it chases after that, and then phagocytoses and eats that cell. And that's one of many kinds of responses that different immune cells have. But that's very different from, say, a molecule in that these are agents that are very smart and very precise. They can move through the body and decide where they should act. They have complex sensing and decision-making capabilities. They can read things like molecules given off by that bacteria and integrate multiple signals. And then they can mediate these very complex, powerful responses. A lot of coordination of a lot of the cytoskeleton and to get that kind of movement and phagocytosis. And so in a way, a cell is much more like a little nanorobot that is able to achieve this kind of much more integrated and complex behavior. And so our interest is in trying to ask, how could we take these sort of nanorobots and change what they do? And just like with if you went and tried to build a Mindstorm robot, that you'd have a bunch of different sensors, you'd plug them into a processor, and then these would link and control different outputs like motors. we sort of are thinking about the cell in the same way that cells have a bunch of molecular sensors or receptors that can detect different things in their environment. These are plugged into various signaling or transcriptional regulatory networks that do the information processing. And then these are linked to different kinds of payloads or actions that occur. And so we know kind of some of the natural structures that do this, but can we actually start to change the programs that they execute by changing what sorts of sensors, what sort of decision-making modules are linked to what responses? Okay, so we've been working on this for a while in sort of simple organisms like yeast, but in the last couple of years, we've really switched a lot of our focus to immune cells. And that's because immune cells represent a really great system to try to look at cellular engineering. So we can actually take T cells, a type of immune cell, from patients. You can now modify them genetically ex vivo, so you don't have to try to target DNA to some place in the body. And then you can expand those cells ex vivo and then transfer them back, just like a blood transfusion. So it actually happens to be a very easy, relatively easy place to do these kinds of modifications. And then moreover, because of the intrinsic functions of these cells, they really can be very powerful. These T cells are the type of cell that can kill an infected cell. So if you could retarget them in particular ways, they could be important for cancer, fighting chronic infection. But then also there are other types of T cells that are good at suppressing inflammation, like T regulatory cells. So they also could be good for treating diseases like autoimmunity or inflammation. Okay. So one of the things that has really opened up this area is the finding that very simple kinds of new sensor molecules can be used to target T cells in a different way. So these are so-called chimeric antigen receptors, and they're essentially synthetic sensors that can redirect T cells to recognize disease cells based on a particular molecule. So So T cells have a so-called T cell receptor that recognizes various complex machinery and then activates the killing pathway. What we can do is we can take part of the inside part that tells a T cell what to do, but then combine it with a new, completely different outside part. In this case, this is an antibody that recognizes a tumor-specific antigen, so a molecule that's presented only on the surface of that tumor. Okay? When you put this kind of chimeric antigen receptor or CAR in, this is actually enough to now instruct that cell to recognize the tumor cell and kill it. Okay? So the way that it's thought to work is you can take those modified T cells, put them back into the body, and then T cells have this amazing ability to infiltrate all the tissues and search around, but they can find those cells, those tumor cells that have that recognized molecule and then execute their killing function. Okay? So so this it seems like science fiction, but what we now know in the last really 4 years or so is that this can really work. So in particular, there are certain B-cell cancers, these blood cancers, where the patients have been resistant to chemotherapy that have been treated with this type of CAR T-cell therapy. And this is one of the first patients that was treated by Carl June and the UPenn group in 2012, Emma Whitehead. And she has been cancer-free since then. So there have been remarkable responses here. About 70 to 80% of the patients have responded with highly durable responses. So this is very different from most cancer therapies. So in other words, T cells can be remarkably effective and are part of the sort of explosion in immuno-oncology today. So, but these CAR T cells still face major challenges. So the success today has still been limited largely to these B-cell or blood cancers. That's going quite well. The first one should be on the market, be approved, hopefully within this year. But solid tumors have been much more difficult. There have been a number of different adverse effects reported in some of the clinical trials that have been done, including several deaths. And there are several different issues with this. One of the ones, and the one that I'll focus on a lot, is that you still have a lot of off-target action. So let's say that, so if you have a molecule that is expressed on the surface of a cancer cell, these designed T cells will kill this. But let's say there's some normal cells that have, even at lower levels of expression, the same molecule. Those cells could be killed. And those are a critical tissue that could lead to lethal side effects. And that, in fact, has been seen. it's interesting that in the case of these B cell tumors, there's a B cell specific antigen, CD19, that's the target. And it turns out that when you give this treatment, you actually wipe out all of the B cells of that patient, the normal ones, the non-cancerous ones. But they're actually expendable. As you give them gamma globulin, they're still okay. So it happens to be that the cross reaction is an expendable tissue, but that's not the case for many of the solid tumors. So in addition, even when they are on target, these cells right now are not very well controlled, and you have this hyperactivation of the immune system that leads to cytokine storm and tumor lysis, these very systemic, strong hyperimmune responses that can, in some cases, be quite severe. So really, what we've learned is that we can engineer T cells to recognize disease, but that that these are really powerful beasts, and that right now, and this is a movie of two T cells actually killing this red cell here, that what we need to really focus on is not necessarily just targeting them, but really having control and precision over these cells. So this is where we've come in and asked, can we take our understanding of systems and synthetic biology and how cells make decisions in order to improve therapeutic T cell engineering? Okay, so what I'm going to talk about is a couple different things in terms of learning how to program cells. If we take some kind of immune cell, the general things that we're interested in are how do we actually put new sensors in these cells to give them the ability to see new things that are associated with disease. And then secondly, how do we actually link this information to new kinds of decision-making circuits where we have some control and can now integrate that information. And then also, how do we actually give the cells new weapons and increased ability to carry out more useful therapeutic actions. Okay, so I'll talk about those three different things. First, let me talk about new sensors and better tumor recognition. So as I said, one of the big problems that I mentioned before was that you could have a CAR T cell that was engineered to recognize a cancer cell that expressed this red antigen, but that if there were bystander cells that shared the same antigen, that you could see potentially lethal cross-reaction with those. And so what we really want to do is to develop systems that allow the cell to use more information in how they recognize cells. How is it that we have this amazing capability today to do things where your cell phone can do face recognition and integrate information that allows it to really identify things with amazing precision? Can we use the same sort of ideas and concepts to increase the ability of a cell to recognize and distinguish a cancer cell from a normal cell? And then a lot of things, and this is true not only for these engineered CAR T cells, but also for any cancer therapy, that a lot of times we focus on one target, or in this case, one receptor or CAR, that for recognizing the target, and we know that that's really limiting, and so it's a situation that's like the three blind men and the elephant, where you get a really misleading view, and the goal is how do you actually get multiple ways of interrogating a tissue or a cell and then integrate that information. Okay? So we've been interested in trying to understand how we could do that, and as I've said before, if you had a tumor with, say, this green antigen or this purple one, you could design T cells that would recognize each of those, but they could cross-react with different tissues. What you'd ideally like to be able to do is to have cells that could integrate information. In this case, I've shown something where this cell potentially would perform sort of a Boolean AND logic where it needed both A and B in order to execute killing. Okay? Okay, so our dream would be if we could actually design cells that had multiple sensors or receptors where you could sense different antigens on that disease, things about the stroma, other things about the microenvironment, and really integrate information across that tissue. Because after all, cancers are a tissue that has sort of different components that contribute to the pathology. But then, of course, we'd like to be able to integrate those signals in the proper way so that we can then control some of the responses that the cells can undertake. So that's where these two guys came in. These are two postdocs, or former postdocs, Leonardo Morsoot and Cole Roybal. They are actually now, he's taken a position at USC, and Cole is going to be a professor at UCSF. But Leo is a developmental biologist and Cole was an immunologist, and they both were interested in new channels for cell-cell sensing and communication. And so they joined together to try to develop some new receptors. And one of the most exciting things that they came up with, which is a really important new kind of engineered receptor that we've been working with called the synthetic Notch receptor, or SynNotch. And it's really a new platform for cell-cell recognition. So Notch is a native receptor that's involved in development in many other places, where the Notch receptor recognizes its ligand, delta, on opposing cells. And this is kind of a mechanical mechanism where pulling of that leads to cleavage, and then the intracellular part is released and goes to the nucleus where it regulates gene expression. What these guys found is that we could actually just take the middle part that mediates that mechanical transduction of Notch and replace the outside with recognition domains like antibodies or other recognition domains, and then replace the inside with orthogonal transcription factors, and then link them to the control of different genes. So essentially, this gives us kind of a universal sensor that allows us to actually recognize what's going on outside the cell, and then use it to turn on a very general thing, which is transcription, gene expression of any gene. We can also put on different sorts of heads on the outside, and at the same time also put on different internal transcription factors and drive different genes with that. So it really isn't just one sensor. These are all, we can get a whole set of different channels, and they're actually all orthogonal to one another, because the one thing that's unique about Notch is that there are no signaling intermediates. So each one is sort of operating on its own, so we can put multiple ones in the same cell and get multiple channels. Okay, so this is an example where we took an antibody that recognizes this B cell antigen CD19, linked it to SynNotch, and if you take sender cells, co-culture them with sender cells that have CD19, what you see is that, and in this case we have GFP as a reporter, we can get very strong transcriptional activation in those receiver cells. So really this is, again, a universal sensor or channel that allows us to give cells new instructions, again, in a very generic form. This is just, for those of you who are more mechanistically minded, the way that we think this works is that that core part of Notch, the Notch receptor, this is one of the initiating proteolytic sites here. It's occluded, it's auto-inhibited by these Lin12 Notch repeats. And so it's thought that when we, either through the native interaction or with this heterologous interaction that we've engineered, apply some sort of pulling force, that exposes the site so it could be cleaved. Okay, so one of the things that's really important about this is that because these synthetic Notch receptors require a signal that's not diffusible and right on the opposing cells, that there's incredible spatial information here. So an example of that is shown here. So this is a case where we take a SynNotch cell and now use it to drive not just GFP, but GFP tagging a master regulator of MyoD, which is a master regulator of muscle fate. And so if you overexpress MyoD, you get these multinucleate muscle myotubes, muscle cells. So if we take these cells and now we plate them, these fibroblasts that have that construct, but now plate them, but only on a spot, a localized spot of the sender cells that have the cognate antigen, then what we see is that after 2 days, these red cells will turn green and become these multinucleate myotubes, but only where those red cells were in contact with the blue cells that had that spatial information. Okay? I won't talk about this in great detail, but one of the things that postdocs Satoshi and then Leonardo have been working on is actually using these sorts of new channels of communication to take cells that don't normally self-organize, but actually try to construct synthetic self-organizing systems that essentially are synthetic organoids, where they start talking to each other and turning on different cascades of signaling and different fates. And we can get, interestingly, a lot of different sorts of self-organizing organoid-like structures from this fairly limited toolkit. This is just an example of a set of cells where we've constructed this artificial developmental program that over the course of about 12 hours forms quite reliably this 3-layered structure of blue, red, and green. So we're actually really excited about the idea, and I won't talk about this too much, but that we can start giving cells, stem cells or other cells, instructions that could allow them to, for example, recognize damaged or injured tissue, and then execute different differentiation events, as well as maybe even the construction of simple kinds of tissues at that site. Just one more side comment, that we can also get the SynNotch molecule sensors to work in neurons. This is just an example where we use neurons, and this is a very simple case where we've co-plated them with K562 cells that have the sender, but we can turn on the strong GFP response. So there is a potential that SynNotch could be a really powerful way to start mapping neuronal connections, or even a way to control what connections are made or maintained, for example. And that just goes to show that the proteases that mediate Notch signaling actually are present in most cell types. OK, so now let me go back to talking about the T cells and how we get them to act more precisely. How do we use SynNotch in these therapeutic T cells, particularly with this goal of trying to engineer T cells that have recognition circuits that can integrate multiple inputs? So one of the first things that we tried to do is to build a very simple AND gate. And in this case, this is a two-receptor system where there's where two antigens are required to get killing. So let me explain this to you. What we have is this is a T cell where we're going to put in the gene for a CAR. This is a killing receptor, but it's not going to be constitutively expressed. But that CAR is going to recognize antigen B. What we do is we also put in a constitutively expressed SynNotch receptor, so one of these transcriptional regulatory receptors, but that recognizes antigen A. Okay? So what that means is that this cell is what we'll call unarmed or unprimed in the basal state because it doesn't have a killing receptor. And so if it encounters a cell with antigen B, it's not going to do anything, okay, because it doesn't have the receptor. If it encounters a cell with antigen A, it will now turn on expression of the CAR, but because there's no antigen B, it also doesn't do anything. But if it encounters a cell that has both antigen A and antigen B, what it'll do is get primed by this one and then kill based on this one. So the idea is that this would require both of those antigens to execute killing. And so we made these and tested them in vitro. And this is just looking at a marker for T cell activation. And it just shows that when we have cognate cells, that target cells that have the dual antigen, we actually get activation. But any of the combinations, any of the single antigen guys do not. So what we really want to do is now see what would happen in vivo. So this is an animation that shows, again, what this issue of having, say, a tumor and some kind of bystander tissue that share these antigens. And the simple CAR T cell would kill the cancer, but it would also be able to cross-react and kill that bystander tissue, which could lead to really severe side effects. In the case where we engineer this novel cell that requires two antigens, in this case, it's using a priming antigen that's recognized by this green receptor that is only present in the cancer cell versus the bystander. But this will turn on the expression transiently as long as it's being stimulated in terms of the first antigen that now allows it to kill the cancer cell. But the idea is that if it enters a tissue where there isn't that priming antigen, that it won't get primed, so it won't kill. Okay. So as I said, that worked in vitro, but we wanted to see whether that would work in vivo in animals. And so what we did is we actually made these tumor models, bilateral tumor models, where we had one tumor that had both antigens, and then a bystander model that only had the single antigen that was recognized by the CAR. And then we injected these CAR T cells and let them go wherever they will and see what would happen. Would they ignore this single antigen side, and would they then would they be able to go into the tumor with both and actually exert that effect? So would they, first of all, be able to, through this kind of transient expression, be able to have a strong enough effect to kill this tumor? And then we were also worried, if that was the case on the other side, would they be able to be primed here and then move here and also kill the other tumor? And so we did these experiments. Cole Roybal did these experiments. And first of all, what we saw is that we have this is the side with the dual tumor. we saw that the T cells actually only proliferated on that one side. So that's a big part of the T cell response, is not just the killing, but they actually have to secrete cytokines and start proliferating. So we saw strong amplification there. And then the tumor clearance study was really quite remarkable, actually much better than we even had hoped, but that on the single antigen tumor side, so the model for the bystander, they continued to grow over the course of, this is 35 days, at a rate that was comparable to what you see if you didn't even add any T cells to this model. But we saw that in all of these mice, the dual antigen tumor was cleared within about 30 days. Okay? So the T cells go in, and they can go wherever they want, but they're getting activated to kill just in one part of the body and not in the other part. So they really are kind of recognizing the landmarks of this particular tissue and showing precise localized activation. So this is actually a movie that shows how spatially and temporally choreographed this kind of response is. What I'm going to show you is a time-lapse movie, and the T cells are in red. They're little. The cancer cells that we use are in gray, and these are the larger cells, and these are cells that have both antigens. And then the T cells are red initially, but when they express, they become primed and express the CAR. That's labeled with GFP. So you'll see them, once they get primed, they're going to turn green, and then you'll be able to tell killing by little bits of cells that kind of come off. So what we see here is that we get these red cells, and they have the SYNACH receptor, so they start binding to multiple target cells here and kind of form these clusters. But then as they sit there, they start turning on this green, which is the expression of the CAR. And you'll see actually it gets quite green. You get these clusters where there's a lot of green in the middle of a bunch of cancer cells. So about 3 hours, they start turning on and becoming primed. And then you'll start seeing a lot more debris coming out, the killing. But by about 10 hours or so, they're highly primed. And then you'll see all these bits coming off, and essentially most of the tumor cells have been cleared. So again, they're sitting there, they recognize that structure, and they're holding on, and then they start killing. But it's a sequence of events that takes, again, quite a few hours to happen. And that's part of the control. Why is it that the prime cells don't go and kill the bystander tumors? And a lot of this has to do with the dynamics of this regulatory program. That we know that you kind of have to go through this 2 steps, that it takes about 6 hours for the CAR to start getting expressed. That's the half-time for expression. And then it takes another 6 hours for the T cells to actually get activated. So that takes, it's really kind of making a decision in a pretty careful way. And that it also turns off if you remove the SYNACH stimulus. The CAR goes down with a half-life of about 6 hours. So that is not short, but it actually is very short within the time window of how long it takes for the cells to clear this tumor. Again, remember, a clearance of a tumor requires cell proliferation and takes weeks, right? So in a way, this is a very short integration time. So it's really kind of control of these dynamics that allows the cells to make very precise and very careful spatial decisions. Okay, so we're really excited about this, and one of the reasons I'm here is we have a great collaboration with Olga and Ben and Ruth, who are here, to try to ask, can we use bioinformatic data to identify what are now sort of combinations of antigens that we could really target with these sorts of cells that would now allow us to discriminate normal versus cancer. And so we have some interesting results that indicate that that it is extremely powerful to go to 2 or 3 antigens, and that probably should be enough in many cases to really distinguish, really tightly distinguish cancer cells. This is an example of just 2 antigens, A and B, where normal samples are in blue and of this particular cancer are in red. And you can see that if you were to look either at just A or B alone, you'd have a hard time picking out a threshold that would distinguish the cancers from the normals. But if you combine them together, you get this this quadrant of the matrix that distinguishes through A and B the cancer. So we're moving forward of trying to use machine learning and other methods to define essentially the actionable signatures that therapeutic cells could use for recognition. So in a way, I think there's a lot of effort to try to distinguish and stratify cancers based on the big data. Essentially, we're trying to do that, but do it through the eyes of a cell. What are the things that cells can recognize, and can we pick those out? OK, so let me now just talk about the last part, which is how do we arm cells with new tools and expand what they can do? And I just want to motivate that by saying that we can target cells to cancers or drugs to cancers. But one of the big problems of cancer is that cancers often are immunosuppressive, so they can actually turn off the immune responses, the endogenous or even these engineered ones. So that in a lot of cases, we think that for effective cancer therapy, we need both, for example, T cells that can recognize the cancer cells, but we also need the ability to overcome the immune suppressive microenvironment. OK, so we wanted to develop ways to give cells new synthetic countermeasures, not just what they sense, but also their response. So when we think about these chimeric antigen receptors that are out there now, essentially what you're doing is you're saying, I'm going to take the T cell receptor and change what it detects. But the response is still largely the same. You have this very complex response of killing, of many cytokines that are released. And what we were interested in is, could we actually edit this or customize this response? Could we come up with more a la carte kinds of responses of what the cells do? And first of all, we're asking, we know that these are complex multi-parameter responses. Some of them are therapeutically good for a certain disease, but some of them can be problematic. Could we edit them in a way so that we emphasize the ones that we really want? And then also, part of why this kind of therapy works is because T cells have this amazing ability to move through the body and infiltrate different tissues and find the target tissues that they're after. And so if we could actually have them do different things or deliver different payloads, then in principle we could take advantage of the sort of modular targeting of these cells. Okay, so what we asked is, could we harness the SynNotch not just to turn on, say, in the case before, another receptor to make a circuit that has two inputs, but rather could we make SynNotch, for example, drive selected sub-responses or even non-natural responses to have synthetic T cell responses, both to sculpt the responses as well as to really harness them as a mobile therapeutic chassis that can harness the spatial migration and flexibility, but also precision of these cells. So just as an example, one of the things that Cole did is if you take a CAR or a TCR, a normal T cell, and stimulate it with the appropriate input, you get a whole set of different cytokines, secreted factors that are part of the immune response. And so if we just plot those here, we can detect the whole battery of cytokines that are released by an activated T cell using the CD19 stimulated CAR. Okay. We can build a SynNotch that also detects the very same antigen, but now is a SynNotch, so it turns on transcription. And we could have it, for example, turn on IL-2, which is one of the cytokines that's produced here, but only IL-2. So we can actually now have this a la carte response that has just that one cytokine, expressed at the same levels to the same antigen, but again is just a singular one. We can also, for example, this IL-12 is something that wasn't expressed natively, but is a really important pro-inflammatory, anti-tumor cytokine, but we can also create cells that will deliver those locally. And that is actually very promising in terms of creating a more favorable anti-tumor microenvironment. So we've done this with a number of different things. We can make things like cytokines that are stimulators. We can also, for example, deliver things like adjuvants, like bacterial proteins that will stimulate, hopefully, the innate immune response, call in other cells of the endogenous immune response that could give you things like essentially a cell-based vaccination. We've also shown that we can produce various factors, either cytokines or other factors that are immunosuppressive, like the cytokine IL-12 or PD-L1, which is a checkpoint inhibitor protein. Those are things that could now lead the cell to for example, treat autoimmunity, where you want to actually dampen the inflammation in a local tissue. And then also, we've shown that we can actually make things that these T cells don't normally make, like antibodies and bispecific antibodies, therapeutic biologics that normally we give systemically, but that could be delivered now in a local way. for custom therapeutic actions. I'm not going to talk about this, but we also have been able to express various master regulators of T cell subfates that drive you towards Th1, Th17, those sorts of things to favor what the outcome is. Some of those are better for cancer. Jasper Williams is a grad student who, for example, made a SYNNOSH T cell that can detect, in this case we detected GFP as an antigen, but that actually produces a cocktail of two different checkpoint inhibitor, anti-PD-1 and anti-CTLA-4, in pretty high amounts in response to the activation. So we could take these things that do have some bad side effects and actually deliver them together and potentially in a much more specific way at the site of a cancer or tissue. One of the things that Jasper did too is, for example, this is using SYNNOSH to express a BiTE. This is a bispecific antibody that recognizes CD19 as a target, but then it also has this engager part that binds to the T cell receptor on a T cell, but it's a secreted factor. We've taken the same sort of bilateral mouse model where we have a dual antigen, something that has GFP here, and then a target tumor that doesn't have GFP as priming. And what we see here is that when we do this, and now in this case it's not expressing a CAR, but this secreted factor, the BiTE, is that we only see strong clearance of the tumor on the side that had the priming SYNNOSH antigen GFP. Okay? So in this case, a secreted factor is produced in a specific way. Okay, so essentially we can think of the T cell now as kind of a local delivery system that can move around, detect where it should be delivering something, and then because we can turn on gene expression, we can deliver cytokines, antibodies, or other things. So we're very excited about how that could be used, not only to fight cancer, but also to do things like immune suppression, or if an antibody is useful for different things. That cells are really, in a sense, a really good way to recognize a tissue and a microenvironment and to actually remodel it by changing what goes on there. And so this could be useful not only for cancer, autoimmunity, but also various inflammatory diseases, degeneration, fibrosis, other things like that. I've told some of the people here that, for example, we actually are really interested in trying to target T cells to the brain, where they could now deliver various things. And we know you can actually get some of these cells in there, and it may be more effective to use a cell as a way to cross the blood-brain barrier than to use molecules directly. So just to give you an idea of kind of where we are hopefully headed, what would be an ideal therapeutic cell for cancer? But this could apply to any kind of other complex tissue-based diseases. And I think the point here is that certain diseases, small molecules are great, but I think in things like cancer or autoimmunity, things where it's really about a complex tissue ecosystem and a system that is harder to recognize, I think that's where cells really can be very useful. But what would the cell therapy of the future kind of look like? And I think if we think about CAR T cells, we of course want to avoid this issue of off-target toxicity with bystander tissues that have shared antigens, but also somehow overcome immune suppression in the cancers. So what are the ways that we could solve these? One is, of course, let's say that you use multiple antigens to prime these cells so that, for example, let's say only in the lung would these cells become primed. They'd recognize the tissue priming, and so they'd only be armed in the lung as opposed to some of those bystander tissues. In addition, let's say you could recognize now the tumor, and that could stimulate the production of things that are pro-inflammatory, as well as things that could block suppression by T regulatory cells or other suppressor cells. And in principle, we don't quite have all these capabilities, but we can imagine being able to link different antigens to these sort of different decision-making circuits and then linking them to things like killing, antibody production, cytokine production in a way to customize both what the cells recognize as well as the kind of response that they launch. And we're very excited with this idea that not just using informatics as a way to stratify and classify diseases, but actually as a way to try to find what are the features that you can really recognize in those tissues, as well as what are the vulnerabilities or issues in the tumor ecosystem that could be targets for what the cell will do. So the idea is that if we combine informatics with systems design and then synthetic biology of being able to build systems that take advantage of that information and those vulnerabilities that really will represent, I think, true precision medicine, where we both combine disease informatics with highly precise, actionable therapeutics. And so we're very excited about trying to do that. But there's a long way to go. But let me thank the people who did the work. This is a movie of a T cell, which just moving around, you can see they're pretty amazing. But I really want to thank Cole Roybal and Leonardo Morsoot, who developed the SYNACH receptors. Cole did all the T cell work on that, but then Jasper did some of the work with delivering antibodies and BiTEs. And then I want to thank folks like Carl June at UPenn, who really introduced me to this field of immune cell therapies. And then, of course, this great collaboration with Olga and then with Dana in the future. So thank you very much. Be happy to answer any questions.